Hello, in today's video, we shall learn in detail about fungus Puccinia graminis. By the end of this video, you'll have a clear understanding about the different types of spores, namely uridospores, tolutospores, basidiospores, pycnospores, acidiospores, and the life cycle of Puccinia graminis, which revolves around the wheat plant and the barberry plant. Let us start with the introduction to Puccinia graminis. Puccinia graminis is an obligate parasite belonging to kingdom Fungi. It is a heteroecious species with two distinct alternate host plants in its life cycle. The wheat plant Triticum vulgaris is called the primary host, while the barberry plant Barberris vulgaris is the secondary or alternate host. The mycelium present in the primary host plant is dikaryotic, that is, it bears two nuclei in each cell. The mycelium present in the secondary host plant is monokaryotic, that is, it bears only one nucleus in each cell. The life cycle of Puccinia graminis is completed only when both the hosts are present. A phenomenon in which a specific pathogen infects only a specific host is called biological specialization. Now, let us understand the vegetative structure of Puccinia graminis. The mycelium is of two types, dikaryotic on wheat plant and monokaryotic on barberry plant. Puccinia graminis shows different types of spores, namely uridospores and tolutospores, which grow on the wheat plant, basidiospores, which grow on soil detached or attached with a dead host, pycnospores and acidiospores, which grow on barberry plant. The dikaryotic mycelium is well developed, branched and septate. The hyphal branches are found in the intercellular spaces of the tissues of stems and leaves of the wheat plant. They give out special knob-like or finger-like structures called haustoria, which penetrate the host cells to obtain nutrition. The transverse septa between the cells have each a central pore to maintain protoplasmic continuity. Cell wall is made up of fungal cellulose. Cytoplasm shows the presence of vacuoles and reserve food materials in the form of oil globules and glycogen bodies. The mycelium is localized. It grows and ramifies near the point of infection. The fully developed mycelium enters the reproductive phase. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us study the reproduction in Puccinia graminis. The dikaryotic mycelium of Puccinia graminis reproduces by sporulation. Two types of spores produced near the surface of the host tissue wheat are uridospores and tolutospores. Uridinyl stage on the primary host wheat plant is formed by the infection of acidiospores, which comes from the barberry plant by wind dispersal, or by the uridospores themselves coming from the infected neighboring wheat plants. The hyphae of the dikaryotic mycelium form a hyphal mass near the surface of the infected organs like stem, leaf, or glumes. Masses of cells are produced subepidermally, which are known as uridia. The uridia gives out slender, vertical, stalk-like hyphae whose tips get swollen up and is known as the uridospore mother cell. It forms a single binucleate oval uridospore. These spores are formed in groups called uridosorus and are seen as reddish-brown pustules. With maturity, the host epidermal wall bursts due to the pressure created by the developing uridospores, which then becomes exposed. The uridospores are stalked, oval, unicellular, binucleate structures. The spore wall is thick with 4 equatorially arranged germ pores. They are dispersed by wind, and during the favorable condition, they germinate on another wheat plant. Let us look into the germination of uridospores and infection of the host. The uridospores, when it falls on another wheat plant during favorable conditions, germinates and produces germ tubes in the form of slender tube through the germ pore. The germ tube enters the host tissue through a stoma. The tip of the germ tube swells up and forms a vesicle-like structure called apressorium. The protoplasm, along with the 2 nuclei, migrate into the apressorium and gets cut off from the germ tube by a septum. A peg-like outgrowth develops from the apressorium, which enters the stomatal aperture and finally reaches the substomatal chamber. The contents of the apressorium reaches the substomatal vesicle. A hypha of the dikaryotic cells develops from the vesicle, ramifies, and branches in the intercellular spaces. The new mycelium produces a new crop of uridospores from the uridia. A layer of binucleate flat cells called basal cells are formed, which elongate vertically and then divide transversally into a lower foot cell and an upper uridospore mother cell. From the latter, daughter cells are formed which swell and form single binucleate oval uridospores, and the lower cell matures into a stalk. Next are the telutospores. Towards the end of the growing season, another kind of spore begins to develop from the uridia in the uridosori. It is the telutospore. This mixed sorus gradually converts into telutosorus. Black raised streaks develop on leaf sheath and also on stem and is therefore called the black spore stage or black rust. The telutospores are bicelled, spindle-shaped, thick, smooth-walled with pointed apex. There is a single germ pore in the wall of each cell. Each cell of the telutospore has 2 nuclei, one of plus strain and the other of minus strain. Towards maturity, the telutospore shows karyogamy. The 2 nuclei in each cell of telutospore fuse to form a diploid nucleus. It represents the reduced diplo phase in the life cycle of wheat rust. This is called the resting period. During favorable condition like high humidity, presence of moisture, low temperature, the Teleotospore germinate on soil detached or attached with dead host to give rise to the basidial stage. Next are the basidiospores. During favorable conditions, the Teleotospore germinate in situ to produce a short, slender, hyaline hypha of limited growth called the promycelium or epibasidium. The diploid nucleus migrates into the epibasidium, undergoes reduction division or meiosis to form 4 haploid nuclei, 2 of plus strain and 2 of minus strain. The epibasidium becomes 4-celled by the development of transverse septa. Thus, 4 uninucleate haploid cells are formed. Each cell develops from projections in the form of short narrow tube, the sterigma. The free tip of each sterigma swells to form a basidiospore, which are small, unicellular, uninucleate haploid structures. Basidiospores are ejected by water droplet method, in which the droplet collects at the base of the basidiospore. A force is created which pushes the basidiospore out and is carried by wind to the leaves of alternate host barberry, which they infect. Haplophase starts with the basidiospores and is present in the alternate host barberry. It consists of primary or haplomycelium and the 2 spore stages, namely spermagonial or pycnidial stage and ascidial stage. The basidiospore germinates on the leaf of the alternate host when the conditions are favorable. The germ tube forms the primary hypha, enters the leaf epidermis, and grows extensively in between the cells to form monokaryotic or haplomycelium. The uninucleate haploid cells are either of plus or of minus strain. Several basidiospores of different strains may infect the same leaf. Fusion occurs at a later stage. Next stage is the spermagonial or pycnidial stage. Haplomycelium forms a dense mat beneath the upper epidermis and is known as primordium of spermagonium. On the lower surface are present primordium of ascidium. From the primordium of spermagonium arise group of hyphae which form a flower-shaped structure called spermagonium or pycnidium. Each spermagonium opens on the upper surface of the host leaf through a pore called ostiole. The wall of the spermagonium consists of 3 kinds of hyphae: spermatiophores or pycnidiophores, periphysis, receptive hyphae. Spermatiophores or pycnidiophores, numerous, slender, short, fine, uninucleate hyphae arise from the cells of the wall. They are closely packed and are called as the spermatiophores. Each spermatiophore by successive division produce many small uninucleate spermatia or pycnidiospore at its tip. The spermatia, when mature, fall into the spermagonial cavity. Periphysis, they are long, delicate, sterile hyphae which develop near the ostiole from the spermogonial wall and later project from the ostiole. Receptive hyphae, they arise from the lateral wall of the spermogonium. They are slender, delicate, cylindrical with blunt, free tips. They may be simple or branched. Now, let us understand spermatization. The mature spermatia exude from the ostiole of spermogonium in a drop of sticky nectar along with the receptive hyphae. Spermatization takes place between spermatia of opposite strains. The transfer of spermatia of one strain to the receptive hyphae of opposite strain is effected by insects. The intervening wall dissolves when the spermatia of one strain comes in contact with the tip of receptive hyphae of opposite strain. Two nuclei lie side by side in a pair and is called a dikaryon. The basal cells of one or more receptive hyphae become dikaryotized. Next stage is the ascidial stage. The ascidial primordia or protoascidium lie beneath the lower epidermis. The basal cells of the primordia become binucleate. They are known as ascidiospore mother cell. They divide producing alternately ascidiospores and intercalary cells. Development of ascidiospores. The ascidiospore mother cell divides transversely to form a large cell and a small cell. The large cell gives rise to a chain of ascidiospores. The lower smaller cell is binucleate, sterile and is known as disjunctor or intercalary cell. Some of the basal cells of ascidium mature into one-celled thick protective layer called the peridium. The ascidium splits open to form a cup-like structure. The ascidiospores fill the ascidial cup, ruptures the peridium and are disseminated by wind. A mature ascidiospore is unicellular, thick-walled, binucleate and orange-yellow in color. They are incapable of germination on the host, that is the barberry plant. They can germinate only on wheat plant under suitable condition. A germ tube emerges out of the germ pore to form the primary hypha which enters the host wheat plant through a stoma. The tip of the germ tube swells to form the appressorium and a vesicle. The hyphae branch extensively and form the dikaryotic mycelium. Hostoria is formed which pierces into the cell to obtain nourishment. The mycelium forms uridospores and the life cycle is repeated. Let us summarize the life cycle of Puccinia graminis, stage 1 in wheat. Primary host wheat plant is infected by ascidiospores from barberry plant. Later, dikaryotic mycelium form uridia which give rise to uridospores. Stage 2 in wheat, Uridosaurus are replaced by the formation of Tilutosaurus that bear tilutospores. Karyogamy results in diploid nucleus formation. It represents the reduced diplophase in the life cycle of wheat rust. Black raised streaks develop on leaf sheath and on stem. This is called the black spore stage or black rust. Stage 3 in wheat, teleospores produce promycelium or epibasidium bearing basidiospores on basidium. Stage 4 in barberry, basidiospores germinate into hyphae with pycnia which bear spermatia and receptive hyphae. Later, a dikaryon is formed when nuclei of opposite strains come close together. Stage 5 in barberry, acidia bearing unicellular thick-walled binucleate acidiospores are produced on barberry. These spores can infect only wheat plant. The graphic representation of the life cycle of Puccinia graminis shows the formation of uridospores and tolutospores, which represent the dikaryophase of the fungus on wheat plant. Haplophase is represented by the basidiospores, which forms two opposite strains of mycelium on barberry leaves. Plus and minus strains of pycniospores are formed. Spermatization takes place, resulting in the formation of dikaryotic hyphae that form acidiospores, which can germinate only after infecting the wheat plant. So today we have learnt in detail about fungus Puccinia graminis, which is a heteroecious species with two distinct alternate host plants, namely wheat and barberry. The formation of different types of spores, namely uridospores, tolutospores, basidiospores, pycniospores, acidiospores. We have also learnt about the life cycle of Puccinia graminis with the help of flowchart, diagrams, and graphic representation. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notifications of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.